In a previous video, I talked about the first three integral membrane proteins in this list. In this video, I'll be talking about the Rh protein and the Rh-associated glycoprotein along with the ABO blood typing system. Now, let us go back to the same figure I featured in the previous video. As can be seen in this figure, the Rh-Rhag complex is part of a macromolecular band 3 complex which stabilizes the membrane. The Rh antigens are expressed as part of a protein complex in the RBC membrane as described earlier. This complex is only expressed in cells of the erythroid line, and therefore, Rh antigens are only expressed in RBCs. The composition of the Rh antigen is unknown, but it is thought to be a tetramer, consisting of 2 molecules of Rh-associated glycoprotein or Rhag and 2 molecules of Rh proteins. Rhag, short for Rh-associated glycoprotein, also referred to as CD241, is actually a member of the ammonium transporter family of proteins. Its exact function remains to be controversial. However, current data suggests that Rhag must be present to direct the Rh antigens to the RBC membrane. If Rhag is missing, as what happens in individuals with the rare Rh null phenotype caused by the deletion of Rhag, none of the Rh antigens are expressed. The absence of the Rh complex, caused by deletions or mutations in the Rhag, alter the RBC shape, increases its osmotic fragility, and shortens its lifespan, resulting in a hemolytic anemia that is usually mild in nature. There are 49 Rh antigens in the Rh blood group system, but only 5 of them are the most important, namely uppercase C, uppercase D, uppercase E, lowercase c, and lowercase e. The first 3 are transmembrane multipass proteins that are integral to the RBC membrane. The Rhd protein is the one that encodes the D antigen and determines if an individual has a positive or negative Rh status. This positive or negative sign is typically stated right after one's ABO blood type. Unlike most cell surface molecules, the Rh proteins are not glycosylated. That is, they do not contain any oligosaccharide moieties, but they are closely related with Rhag as described earlier. The Rh antigen has no spontaneous agglutinins. Anti-D, anti-C, anti-E, and anti-lowercase e antibodies are described, but they require previous exposure for formation. These antibodies have all been involved in hemolytic transfusion reactions, particularly delayed reactions. Routine blood typing for Rhd status in both blood donors and transfusion recipients has reduced the incidence of transfusion reactions caused by anti-D. But sensitization to other Rh antigens can be a problem in transfusion medicine, particularly in patients with sickle cell anemia who typically require frequent blood transfusions. From this graph, you can see that the Rhd positive phenotype is overwhelmingly more common than the Rhd negative phenotype. Among those who are Rh negative, about 15% are Caucasians, about 8% are Blacks, and only 1% are Asians. Now, from this table here, you can see that Rh group inheritance appears to follow Mendelian genetics. Basically, each Rh gene locus has its own set of alleles. Now, the Rh factor genetic information is inherited from both parents, but it is inherited independently of the ABO blood type alleles. Each biological parent donates one of their 2 Rh alleles to their child. As will be discussed later, inheritance of ABO blood types also follow this mode. Now, this slide, to which I credit the New York Blood Center, shows here a diagram indicating that the Rhd protein is also a multipass protein. Additionally, you can have other Rhd phenotypes such as partial D, weak D, and the del phenotype. The partial D phenotype is usually caused by the creation of a hybrid Rhd and Rhce protein. The hybrid protein is similar enough to the usual Rhd protein, so much so that partial D patients should not receive Rhd positive blood. If a person with partial D phenotype encounters the complete D antigen on transfused RBCs, they may form anti-D and suffer from a transfusion reaction. In practice, however, people with partial D are difficult to identify. The weak D phenotype, on the other hand, is typically caused by a single amino acid switch in the transmembrane region of the RHD protein. This disrupts how the RHD protein is inserted into the RBC membrane, reducing the level of expression of RHD. For that matter, individuals with the weak D phenotype can be able to receive RHD positive blood. The DEL phenotype represents a very weak form of D variant detected only by absorption and elution techniques. DEL phenotype individuals are very often mistyped as RHD negative, leading to alloimmunization following transfusion or pregnancy. When it comes to blood transfusion, anyone who is Rh positive can receive blood from someone who is Rh negative, but those with negative blood types cannot receive from anyone with a positive blood type. Rh antigens are highly immunogenic. Individuals who do not produce the D antigen will produce anti-D antibodies if they encounter the D antigen on transfused RBCs, causing a hemolytic transfusion reaction on subsequent exposures. For this reason, the Rh status is routinely determined in blood donors, transfusion recipients, and in mothers-to-be. There are no preformed anti-Rh antibodies in one's blood normally. After the first exposure, however, to Rh positive blood, the immune system of an Rh negative individual reacts and starts making antibodies. Basically, a transfusion reaction does not occur the first time a Rh negative patient is exposed to Rh positive blood because the Rh negative patient has not yet produced anti-D antibodies. Subsequent transfusions of an Rh negative individual with Rh positive blood would then result in agglutination of erythrocytes and hemolytic transfusion reactions. Of note, Rh antibodies rarely activate complement. They bind to RBCs and mark them up for destruction in the spleen in a process called extravascular hemolysis. Now, something similar happens when an Rh negative mother conceives an Rh positive baby. Rh incompatibility occurs when a pregnant woman whose blood type is Rh negative is exposed to Rh positive blood from her fetus, leading to the mother's development of Rh antibodies. During pregnancy, though, the placental barrier basically blocks off any possibility of mixing of fetal blood with maternal blood. However, during delivery, the Rh positive baby's blood cells can enter the mother's bloodstream. The invading Rh positive blood cells then cause the production of maternal Rh antibodies, which can persist for a long time. Now, when the mother again gets pregnant with another Rh positive baby, the maternal Rh antibodies now have the potential to cross the placenta and attach to fetal red blood cells, resulting in fetal RBC hemolysis, leading to a condition called erythroblastosis fetalis. In severe cases, an intrauterine blood transfusion for the fetus may be required to correct the anemia. For this reason, as was mentioned in the previous slide, the Rh status is routinely determined in blood donors, transfusion recipients, and in mothers-to-be. We now move on to the ABO blood group system. Now, this blood group system is actually the most important, with the Rh system coming as a close second. The loosely used term blood type actually refers to a defined set of red blood cell antigens or blood group substances controlled by a genetic locus having a variable number of alleles, that is, A, B, and O. The term blood type refers to the antigenic phenotype, usually recognized by the use of appropriate antibodies in the laboratory. Now, the ABO antigens are complex oligosaccharides that are actually present in most cells of the body and in certain secretions, not just the RBCs. For RBCs, these oligosaccharides appear to be mostly present in glycosphingolipids, whereas in secretions, the same oligosaccharides are present in glycoproteins. Now, unlike with Rh incompatibility, agglutinins form with ABO incompatibility, and this I'll be showing in a subsequent slide. Also, hemolytic transfusion reactions occur immediately upon first exposure to the wrong ABO blood group, unlike with Rh incompatibility, wherein the hemolytic transfusion reaction occurs only on subsequent exposure to the wrong Rh blood group. When ABO incompatibility occurs in the fetus or in the newborn, hemolytic disease of the newborn results. The usual setup here is that the mother is type O, while the baby is either type A, B, or AB. As will be explained further later, type O individuals have naturally occurring anti-A and anti-B IgG antibodies, which can cross the placenta and hemolyze fetal RBC. Now, as far as worldwide distribution of each ABO blood type is concerned, we don't really know for certain. Among the various references looked up, blood type O consistently appears to be the most frequent, with blood type AB the least frequent. In this table, we see the percentages stratified according to RHD status. Keep in mind, though, that these are worldwide figures. Prevalences of each blood type are heterogeneous, depending on what regions in the world. Among the entire population of blood type B individuals, the prevalence appears to be highest in Central Asia and lowest among the indigenous peoples of the Americas and Australia. Among the entire population of blood type A individuals, the highest frequencies are found in small, unrelated populations, especially the Blackfoot Indians of Montana, the Australian Aborigines, and the Lapps or Sami people of northern Scandinavia. The A allele apparently was absent among Central and South American Indians. Finally, blood type O is particularly high in frequency among the indigenous populations of Central and South America, where it approaches 100%. It also is relatively high among Australian Aborigines and in Western Europe, especially in populations with Celtic ancestors. The lowest frequency of blood type O is found in Eastern Europe and Central Asia. I mentioned how agglutination is immediate in ABO incompatibility and not in RH incompatibility. When we are talking about the ABO blood typing system, the ABO blood type a person has refers to the antigen present on their RBC surface. Blood type A individuals have the A antigen on their RBC surface, but have preformed anti-B antibodies in their plasma. Similarly, blood type B individuals have the B antigen on their RBC surface, but have preformed anti-A antibodies in their plasma. Blood type AB individuals have both A and B antigens on their RBC surface, and neither preformed anti-A and anti-B antibodies in their plasma. Lastly, blood type O individuals have neither antigens on their RBC surface, but have both preformed anti-A and anti-B antibodies in their plasma. In the laboratory, ABO blood typing is performed by adding 2 drops of blood in a single slide. Anti-sera A is added to 1 drop, while anti-sera B is added to the other. If blood agglutination occurs in the blood drop to which anti-sera A was added, then the person is blood type A. If agglutination occurs where anti-sera B was added, then the person is blood type B. If agglutination is seen in both drops of blood, that is, agglutination was observed with addition of anti-sera A and anti-sera B, then the person is blood type AB. If agglutination occurs in neither, then the person is blood type O. Now, of course, in standard laboratories, 3 drops of blood per slide is frequently needed because blood typing is carried out with both the ABO and RH systems, and the other minor blood groups whenever indicated. Now, with all of this information in mind, we now understand why blood type O negative individuals are regarded as universal donors, while blood type AB positive individuals are regarded as universal recipients. Now, you may be wondering why type O blood can be given to other ABO blood types when individuals with type O blood have preformed antibodies in their plasma. In the hospital setting, unless the situation calls for giving fresh whole blood, blood is more frequently transfused to a patient as packed red blood cells or packed RBCs. Packed RBCs are prepared from a unit of whole blood by centrifugation and removal of most of the plasma. Let us now move on to different ABO antigens. As I said earlier, the A, B, and O red blood cell antigens, also called blood group substances, are controlled by a genetic locus having a variable number of alleles. The blood group substance found in type O persons is called H substance, sometimes referred to as O substance, which, as you'll see later, gives rise to both A substance and B substance. Type O persons have an H gene locus that codes for the enzyme fucosyltransferase. This enzyme adds the monosaccharide fucose onto the terminal galactose residue of the H substance precursor to produce H substance. Now, type A individuals have an A gene that encodes for a transferase enzyme that adds a terminal GalNAc onto H substance, thus converting it to A substance. Type B individuals, on the other hand, have a B gene that encodes the transferase enzyme that adds a galactose residue onto H substance, thus converting it to B substance. Now, with type O individuals, however, a frame shift mutation has occurred such that an inactive glucosyl transferase enzyme is encoded. Anti-antibodies are directed against the terminal GalNAc that was added onto H substance, while anti-B antibodies are directed against the galactose residue that was added onto H substance. Now, for type O individuals, however, since they are unable to add neither sugar residue onto H substance, they have no antigen that both anti-A and anti-B antibodies can target. Now, this diagram here embodies the same concept I discussed earlier. But here, I would like to reiterate that with type O individuals, a frame shift mutation has occurred such that an inactive glycosyl transferase enzyme is encoded. Now, let's try to understand the mode of inheritance of ABO blood groups. Codominance in genetics means that neither allele can mask the expression of the other allele. The human ABO blood group system exhibits codominance. The system consists of three alleles, A, B, and O. Both A and B are dominant in relation to O, and therefore, blood group A can have the genotype AA or AO, and blood group B can have the genotype BB or BO. However, neither A nor B shows dominance over the other, and therefore, individuals with the genotype AB have the phenotypic characteristics of both blood group A and blood group B. Now, which offspring gets which allele follows the basic Mendelian pattern, as is depicted in this figure. In this table, we have different ABO blood group phenotypes and genotypes of the father and mother, along with the corresponding possible and impossible blood groups of the resulting offspring. In cases of questioned paternity, ABO blood typing can be used to exclude a man from being a child's father. For example, a man who has type AB blood could not father a child with type O blood, because he would only pass on either A or the B allele to all of his offspring. Now, recall from earlier, I said that type O persons have an H gene locus that codes for the enzyme fucosyl transferase. Recall that this enzyme adds the monosaccharide fucose onto the terminal galactose residue of the H substance precursor to produce H substance. Now, there's a rare blood type wherein affected individuals are unable to express H substance. This is because these individuals have both alleles to the H gene that are mutated and thus are unable to encode for functional fucosyl transferase. Because of this, the H substance precursor is not converted to H substance. For that matter, individuals who have the uppercase H, lowercase h genotype are still able to yield adequate levels of H substance because they are still able to encode functional fucosyl transferase enzyme. It is a different story, however, if an individual has both problematic alleles to the H gene, as I described earlier on in this slide. Now, this blood type has been named the Bombay phenotype because it is mostly found in the Indian subcontinent and parts of the Middle East, such as Iran. Since these individuals are unable to produce either A substance or B substance because they are unable to produce H substance in the first place, people who have Bombay phenotype can donate red blood cells to any other member of the ABO blood group system, as long as they are also Rh compatible. But they cannot receive blood from any member of the ABO blood group system, which always contains one or more of A, B, or H antigens, but only from other people who also have the Bombay phenotype. Now, as part of independent learning, you are required to read this article, which I have uploaded in Canvas. This article is about the potential for engineering the transferase enzymes that I discussed earlier in order to generate antigen-null blood from donors of various ABO blood types. Now, this modality finds promise and is especially potentially life-saving for patients with uncommon blood types who are in dire need of blood. I end part 4 of this video series here. Please proceed to the 5th and last video, which will be on white blood cells.